Hello everyone, my name is Gore Pranita Satish. I am a student of t.patil.clg.of.pharmacy, Usmanabad. I am studying in first year B Pharmacy. Today we will discuss the biosynthesis of purine nucleotides, in which included points deneosynthesis of purine nucleotides, precursor of the members of purine ring, and its process of biosynthesis of purine nucleotide, its significance, salvage pathway and their enzymes and significance. Today, there will discuss the history of purine nucleotides. In short, DNA structure. DNA structure we study in 12th standard. That's like the nucleotides are base pairs. Like purine and pyramids are the basis of nucleosides. Adenine and guanine are two purine bases. There will be discussed in this lecture. Uh, Deneosynthesis. It is a method by which purine ring synthesized with nucleotide attached to the ribose sugar generated from HMP pathway. Active D-ribose 5-phosphate as the starting material for synthesis of purine. The first idea of purine biosynthesis in this cell was studied by John Buchan in 1948 by radioactive tracer technique in bird during study of uric acid. After in 1950, Dr. Buchan and Robert G. Greenberger together studied detail about purine bases, purine basis biosynthesis with radio labeled method. As 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 per the study, the sources of different atoms in purine skeleton were identified, which are like as precursor of the members of purine ring. N1 is contributed by nitrogen of aspartate. N3 and N9 arise from amide nitrogen of glutamine. C2 and C8 originate from the format. C6 is embedded from respiratory carbon dioxide, that is from bicarbonate. C4, C5 and N7 are taken up from glycine. Here is the structure of purine nucleus. Then we will discuss the whole process of purine biosynthesis of purine nucleotides. There is a whole process like it is divided into step 11. In step 11, the process like the first metabolic synthesis starts with inosine 5 monophosphate. Step first, activation of ribose 5 phosphate and formation of 5 phosphoribosal alpha pyrophosphate. In short, we call the PRPP. Alpha D ribose 5 phosphate is activated by ATP and converted to 5 phosphoribosal alpha pyrophosphate with the help of a ribose 5-phosphate phosphokinase enzyme. There will be product is also precursor for pyrimidine synthesis and some amino acid synthesis like tryptophan, histidine, etc. Step second, acquisition of N9 atom of purine. The pyrophosphate in C1 and 5-phospho-ribosol alpha pyrophosphate are replaced by an amide nitrogen group of glutamine and form of 5-phosphoribosol 1 amine with the help of amidophosphoryzole transferase enzyme. This formation step is known as committed step. The reaction contributes N9 atom of purine from glutamine. Step third, acquisition of C4, C5, N9, N7 atom of purine. Carboxyl group of glycine is combined with amino group of beta 5-phosphoribosol amine to form glycinamide riboside with the help of a glycinamide riboside synthesis enzyme. Step 4, acquisition of C8 atom of purine. Amino group of glycinamide riboside is formulated with N10 formyl transhydrofolate and form of forming glycinamide riboside with the presence of glycinamide riboside transamylase enzyme. Step 5, acquisition of N3 atom of purine. There will be amide nitrogen of second glutamine is added to formoglycinamide riboside in an ATP dependent reaction to form formylglycinamide riboside with the help of a formylglycinamide riboside synthesis enzyme. Step 6, purine imidazole ring formation. An ATP dependent ring closing reaction takes place and forms 5 amino imidazoyl riboside with the help of a 5 amino imidazoyl riboside synthesis enzyme. Step 7. Acquisition of C6 atom of purine. 5 amino diazole riboside further react with bicarbonate with the help of a 5 amino imidazoyl riboside carboxylase enzyme to form carboxyamino imidazoyl riboside. This reaction is ATP dependent and named as carboxylation. Step 8. Active acquisition of N1 atom of purine. Amide bond is formed with C6 when aspartate is added and formed 5 amino imidazoyl 4, that's called N-succinylcarboxyamide riboside in presence of 5 amino imidazoyl 4, that's called N-succinylcarboxyamide riboside synthesis enzyme. This reaction is also ATP dependent. Step 9. Elimination of fumarate. Fumarate group is cleaved off from 5 amino imidazoyl for N-succinyl carboxyamide riboside to from 5 amino imidazoyl for carboxyamide riboside in presence of adenosine enzyme. Step 10. Acquisition of C2 atom of purine. Amino group of 5 amino imidazoyl 4 carboxyamide riboside reacts with neoformylase formetyl tetrahydrofolate to form 5 imidazoyl 4 carboxyamide riboside in the presence of 5 amino imidazoyl 4 carboxyamide riboside transformylase enzyme. This reaction is also known as formylation. Step 11. Cyclation to form IMP. The larger ring of 5 amino imidazoyl 4 carboxyamide riboside is enzymatically closed to form inosine monophosphate. That's in short called IMP. With the help of inosine monophosphate cyclohydrolase by release of water molecules. This is a reaction is called a catalytic reaction. Then we will discuss the significance of this process. It occurs in the liver. Ribose 5-phosphate is the starting compound. Inosine 5-monophosphate is the first purine synthesized from ribose 5-phosphate. It is a 6-membered ring. In this whole reaction, 6 ATP is utilized. Then we will discuss the regulation of purine nucleotide by denosynthesis. Purine biosynthesis is regulated by feedback inhibition. The inhibition is in the first step. It is a committed step by which generally irreversible. The different mechanism by which it is regulated are as follow. It is a structure of these irreversible reactions. There will occurs regulation of purine synthesis. Then we will discuss the salvage pathway. The salvage pathway is the only one pathway by which nucleotide synthesis occurs in brain and bone marrow tissues. The the it the denosynthesis does not occur in brain cells and leukocytes. Hence, does not occur in brain cells and leukocytes. It involves synthesis purine nucleotides from pre-purine bases salvaged from dietary sources and tissue breakdown. This pathway is there for energy saving pathway as compared to denosynthesis. The enzymes of this pathway adenine phosphorybase transferase, that's in short called APRT, and hypoxanthine guanine phosphorybase transferase, that in short called HGPRT. The function of these enzymes are as follow: adenine 5-PRPP, it's undergo the enzyme adenine phosphorybonyl transferase with the help of AMP plus PPI. Xanthine plus 5-PRPP with the help of HGPRT, XMP plus PPM. Hypoxanthine plus 5-PRPP in HGPRT with the help of IMPN plus PPI. Guanine plus 5-PRPP. It gives HGPRT, GMP plus PPI. There will be new synthesis of purine biosynthesis salvage pathway in short of this is the structure of salvage pathway and their significance are as follow. Salvage pathway occurs in extrahepatic tissue like brain and bone marrow. This pathway is a recycling of products of nucleic acid breakdown. It occurs from pre-adenine and guanine bases. Uh, this pathway is energy saving pathway. Then de novo pathway. De novo pathway, de novo pathway. After this pathway is also energy saving pathway. Um, if purine bases are not salvaged, then de novo pathway we will be activated to provide missing nucleotide by two ways. Decrease in salvage in purine, we will make more PRPP available to stimulate de novo pathway. It also decrease the nucleotide that's like AMP, IMP, GMP available to inhibit de novo synthesis. Here is the today's lecture is over. Thank you for listening this seminar. Thank you Rajman ma'am. Thank you student.